I want to welcome everybody to our Breakthroughs in Cancer, and including those that are on Zoom and listening to us on Zoom today. So I'd like to introduce myself. I'm Melissa Bondi, and I'm the Associate Director for Population Sciences, and I'm also the Chair of the Department of Epidemiology and Population Health. So I'm really happy to be here today to introduce this seminar speaker, Dr. Cornelia Ulrich from the Huntsman Cancer Institute. So our Breakthroughs in Cancer seminar series is one of the highest profile seminar series for translational and clinical scientists in oncology. We bring national experts to Stanford campus each month to discuss innovations across the spectrum of cancer research. So now I want to introduce our speaker today, and it's really my pleasure and honor because Dr. Ulrich and I have known each other probably for, I won't say how long our careers are, but probably for most of our career. We followed in many of the same epicircles, and it's really been a pleasure for me to get to have her here today and to take a walk around campus and show her around. And so we had a really great dinner last night, and she's gotten to meet some of our faculty, our students, and we had a really, I hope that you got to have good flavor for what it's like here at Stanford. So Dr. Ulrich is the Executive Director of the Comprehensive Cancer Center at Huntsman Cancer Institute. She is also the John M. and Karen Huntsman Presidential Professor in Cancer Research in the Department of Population Health Sciences at the University of Utah. And I want to point out, first of all, there's very few women that are cancer center directors, maybe more now, but also very few women and PhDs that are cancer center directors. So this is really a major accolade of Dr. Ulrich's accomplishments, and I just wanted to highlight that for all of you to know. She's also the Chief Scientific Officer and Cancer Center Executive Director. She oversees the Huntsman's academic consortium of more than 260 research teams, and she is, as I mentioned earlier, a cancer epidemiologist whose research focuses on lifestyle and biologic factors in cancer prevention and cancer prognosis. She specifically focuses on colorectal cancer, and she leads an interdisciplinary team of scientists related to prevention, epidemiology, prognosis, and survivorship. And she's the lead PI on a study called ColoCare, which she'll talk about today. And she also has multiple R01 type grants related to colorectal cancer. Her lab has performed landmark research on colorectal cancer prevention and studies on biomarkers, aspirin, and their connections between obesity and physical activity in cancer prevention and survivorship. She's conducted several clinical trials. She's the author of more than 480 publications and has served as principal investigator on more than 26 competitively funded research studies and clinical trials. She's a former Fulbright Scholar, member of the Board of Scientific Advisors of the National Cancer Institute, and has served on numerous advisory committees and leadership roles, including the AACR and the AACI, which is the Association of Cancer Institutes. She's also European Association of Cancer Research. She spent a lot of time in Heidelberg at the DKFZ and was there before she came back to the United States. She has received numerous honors, including Utah's Governor Medal for Science and Technology. Dr. Ulrich, we're really excited that you're here and look forward to your talk.
Well, I'm thrilled to be here today and enjoy the opportunity to get to know Stanford a little bit better and Stanford Cancer Institute, and also special thanks to both Melissa and to Steve for inviting me to give this important and highly valued seminar. So let's just say briefly, this is Huntsman Cancer Institute. We are 25 years old. We have currently 1 million square feet, and we have about half of our faculty in these 5 buildings. as well as another half in the surrounding buildings of the University of Utah. Just like you guys, we're starting a new cancer center, a campus that will encompass both clinical and research care, and that will be happening, like groundbreaking is in a month. So if you want to know more, we can talk about that. You see the venue is slightly different, and that's important. We're right next to the mountains, and we have a special need to serve populations that are housed in those mountains. So first of all, I want to say I actually have no meaningful disclosures, except for that I oversee all the research activities at our Cancer Institute. So what I'm not going to talk about today is the beginnings. It's like my work in genetics, folate, genetics, pharmacogenetics, mathematical modeling, NSAIDs in cancer prevention and precision prevention. But what I would like to share is what we've been doing in transdisciplinary team science in colorectal cancer survivorship. I'll focus especially on one topic, which is the energy balance as a driver of cancer outcomes. And I'll talk also about early onset colorectal cancer, which is a major challenge around the world. And I'll do a little bit more work telling you about exercise interventions in cancer patients, and finally share what we are doing at Huntsman Cancer Institute to overcome what we call as distance as a disparity. Obesity. We all know it's a major public health burden in the US, but still, I'm always shocked when I look at these numbers. 42.4% of adults in the United States in 2017 were obese, and another 10% severely obese. This is before the pandemic. During the pandemic, this percentage has increased even more because we know that 42% reported they gained weight with an average weight gain of another 30 pounds. And in addition, we have more than 14 million US youth being obese. Now, of course, with GLP-1 inhibitors, this may change, but right now we have a tremendous problem. And I was part of a working group at the International Agency for Research on Cancer, which demonstrated that 13 types of cancer are convincingly associated with obesity. Now, many of you in the room here will be basic scientists and wonder, well, what is it about obesity? There are a number of biologic mechanisms, and if you want to know more, there's a review that we put out in Nature Reviews in 2018. A lot of it is still true. There are many important mechanisms, and as this graph here illustrates, is the red lines are those that are inversely associated. Obviously, caloric restriction reduces obesity, and physical activity with a lesser thick line to a lesser extent. And these other factors then, these associations are described by lines that are positive associations. So obesity is the major driver of systemic inflammation in the body. It affects the insulin axis, it affects sex hormone levels, adipokines, etc. But there are also other mechanisms that I'm not going to talk about today, including oxidative stress, DNA repair, etc., immune function. I've done a lot of research on all of this, including in intervention studies, but there's not enough time. So let's focus on some central themes, which is inflammation, adipose-derived factors, and the gut microbiome. The study that I want to talk about mostly is the ColoCare study. We came together as a team in 2009 and really wanted to address colorectal cancer prognosis from a comprehensive angle. It's now, because of various moves of investigators, including myself, an international multi-center cohort of people of colorectal cancer. More than 4,300 have been recruited, and I want to point out that it is an open resource. It's funded by a UO1. If you're interested in learning more, there's a website as well as a study design publication. And what you see us doing here is holding up our hands as a 10, which means the first meeting after COVID where we were celebrating our renewal at the first time submission with a perfect score of 10. And that was in parts because we were so good in data sharing. What does this look like in terms of where the world is? Like, here's the team across the United States and Germany. We have now 6 US sites and 1 German site. And as you see, they are nicely distributed across the country. I mentioned that we wanted to set out to understand the many factors that can affect colorectal cancer survivorship. And in order to do that, we started out by saying, let's try to really get all the biospecimens we possibly can at baseline. So that means pre-surgery blood, tumor and normal tissue, as well as 2 types of adipose tissue: urine, feces, saliva, CT measurements, anthropometrics, and questionnaires. And then, as many times as our patients were willing to participate, continue with questionnaires on symptoms, quality of life, and current health habits, and additional collections of biospecimens, because we anticipated that some of these would change over time. And for example, we have done work looking at multiple assessments and multiple time points of metabolomics in relation to recurrence. So this is the kind of work that could be done, but outcomes could be not only recurrence and survival, but also symptoms, quality of life, treatment toxicity, etc. Now, I should say that with that renewal came a budget cut, because we also had now 7 sites instead of 5. So we're focusing on a subset of these assessments. Not everything is available on everybody, but we have a lot of cool data. Okay, what are we doing for energy balance biomarkers and other related factors within the ColoCare study? Physical activity is measured with accelerometry. For adiposity, I want to talk a little bit about these kind of pictures. So these are CT scans of people at the level L3 or L4. And what you see is this person on top has a very slim outer layer. This is the subcutaneous fat, whereas this person has a lot of subcutaneous fat. And this person has a lot on the inside. This is visceral adipose tissue, and this person has less on the inside. I'll tell you a little bit more about why we care about that. We have done here metabolomics. We looked at a number of different outcomes, and we also looked at the interaction between the adipose tissue and the tumor that is growing in its adjacency. So adipose tissue was long seen just as a storage organ for fat, and it's not. It's actually a very bioactive organ in our body, especially if you have a lot of it. And we know now that it's made up of adipocytes, macrophages, lymphocytes, etc., secretes a number of factors, including leptin and cytokines. And some of these are clearly predisposed to interact with the growing tumor, especially with respect to inflammation and angiogenesis. Now, starting out looking at these different types of adipose tissue, we started with gene expression analysis. And so Mariam Haffa, who is one of my former doctoral students, looked at what does this look like and what do we care about here? Is this like the visceral adipose tissue or subcutaneous adipose tissue? These are from the same patient, and you immediately see that gene expression profiles are clearly clustering very differently. The next doctoral student then worked on metabolomics analysis, a collaboration with the West Coast Metabolomics Center, Oliver Fien. And what you mostly see here in red is that these metabolites are all elevated in visceral adipose tissue, and most of them are red. There's only a few that are higher in subcutaneous adipose tissue. So what this tells us is that there's a higher abundance of amino acid nucleotides and carbohydrates, and in an integrated analysis between the metabolomics and the transcriptomics, it also became clear that not only do we have more arachidonic acid in VAT compared to SAT, but a real driver towards inflammation and the release of prostaglandins in visceral adipose tissue. And this has been confirmed in a number of other studies. The visceral adipose tissue is really what's more metabolically active and is also a driver to a greater extent of diabetes, for example. Now, the next question, the next doctoral student is like, how does this matter? Does this matter with respect to what we see systemically? And does it matter how much visceral fat people have? This is a study within our ColoCare patients. and they had measured visceral and subcutaneous fat area, so the amount of fat. And we had a panel of biomarkers both on inflammation and angiogenesis, all of them measured prior to surgery, which is critical for these biomarkers. What you see here in this heat map is here's the subcutaneous fat area and the different markers of inflammation and angiogenesis. There's not much going on. Here is the visceral fat area, and the correlations become stronger. And then if we form the ratio of visceral fat area to subcutaneous fat area, this is where the correlations become even more pronounced. And for the very first time, we could actually show that an angiogenesis marker in the blood was elevated among patients who had greater amounts of visceral fat compared to subcutaneous fat. Now, why do we care about those biomarkers? Next question is, are they related to survival in our colorectal cancer patients? Here we have now more than 400 stage 1 through 3 ColoCare patients, the same angiogenesis and inflammation biomarkers as well as some adhesion biomarkers measured, and 65 deaths. And what Jenny Ose, who's now professor in Hanover in Germany, found is that there is like substantial increases in risk of death with SICAM for both colon and rectal cancer, and significant heterogeneity in risk of death between colon and rectal cancer for VEGF-D, an angiogenesis marker, and also significant heterogeneity for CRP and SAA, both unspecific biomarkers of inflammation. And this is a tremendously increased risk for colon cancer with CRP and SAA in the highest tertile. So one of the questions is, aside of these associations, are there distinct mechanisms both for disease etiology and progression for colon and rectal cancer that we need to consider? Obviously, a colon cancer, the recurrence mostly happens in the liver, whereas rectal cancer typically has recurrences locally, and that could be like a key factor in driving these differences. So the next question was, okay, well, we know that there are systemic influences of adiposity that change the inflammatory profile of a patient, but are there also paracrine influences with adipose tissue, or perhaps direct crosstalk reflecting a true adipose tissue tumor microenvironment? So as background, several obesity-associated cancers, including those of the colon and rectum, arise within or in close proximity to adipose depots. And if we talk about breast cancer or prostate cancer, it's within the same organ. But the colon is surrounded by metabolically active vat, but it lacks direct interaction by the presence within the same tissue. So when we talk about tumor-adjacent vat, it's within 10 centimeters of the colon. We have recently received an R01 with Steve Hursting and Chetan Roper, and that is a transdisciplinary study, which I'm most excited about, because in humans we can get only so far, but we have parallel studies of mice and humans, so the experimental work as well as the human relevance within the same grant. There was an initial paper published by Andriana Holovadi where we showed some evidence of crosstalk. You're welcome to look that up, but we now have more data, and I just want to share some of that with you. So Vicky Bandera, doctoral student, is or has just recently presented at the AACR a basic question whether obesity status actually is associated with altered gene expression pathway in colon tumors. And she identified through IPA that there were multiple activated, inhibited, and disease-related pathways among those who are obese versus non-obese. The overall signals were that those with a higher BMI had pathways associated with more aggressive tumors, such as increased cell movement and decreased cell death. I'm going to share with you some not yet published data to look at direct crosstalk in the adipose tissue colon tumor environment. Tengda Lin, another doctoral student, is looking for the paired tumor adjacent and tumor gene expression here is the initial just basic correlation of pathways in the adipose and tumor tissues to figure out, for example, do we have a greater secretion of IL-6 corresponding to greater upregulation of receptors in the tumor. And these multi-database analyses that he then conducted linked adipose-derived ligands with tumor receptors, and those suggest increases in metabolic, inflammatory, differentiation, and proliferation pathways. So there is like now more evidence for this direct crosstalk and tumor microenvironment. So with that, I'm going to leave this alone for a minute, the inflammation, and step over to the microbiome. So microbiome, everybody knows a lot about it, but just to remind ourselves, so we have in our bodies about 30 trillion human cells, but our microbiome is an estimated 39 trillion microbial cells. And so these little bacteria, viruses, and fungi live on us and in us. And here you can see there is like a range of interactions that range from like adverse in terms of pathogens to symbionts that have been mutually co-developing with us. And then we have like commensals and others. So some are good players, some are bad players, but it's so complicated to assess them. I'm not the expert on everything of this, so I just want to say that from the beginning. But I'll share a little bit of data with you that we have looked at. So the main measure I want to talk about is alpha diversity. Alpha diversity of the microbiome is a measure of richness, which means how many different species, and evenness, how many of each species. And it's expressed at either a Shannon or a Simpson index. And this is, if you think from an evolutionary perspective, it basically, the higher level of alpha diversity means like an improved community that will be relevant if there are challenges happening to your system. Now, in relation to what evidence has existed on energy balances, athletes have decreased alpha diversity, we can talk about that, increased abundance of certain strains. Obesity is associated with decreased alpha diversity and decreased abundance of Bacteroidetes and Firmicutes. And for colorectal cancer, very consistently now we see decreased alpha diversity and increased abundances of some bacteria, including Fusobacterium nucleatum, or Fuso, which has consistently emerged as a player in helping to drive colorectal cancer. Now, there are also some good players that are, when you have less of them, you have increased colorectal cancer risk. And one of them is Faecalibacterium prausnitzii, and that is related to short-chain fatty acid-producing microbiota. So just remember, better alpha diversity is better, Fuso is bad, and some of these good players are relevant as well. There's a lot of data we have, including now on cachexia, that I'm not able to show, but let's talk about what it looks like for energy balance. So Caroline Himbert, who is now faculty in Utah, looked at 4 different groups. She looked at patients who are either active or inactive, and normal weight or overweight. And these are the 4 different groups. She showed that there were significant differences in gut microbiome diversity and abundances by physical activity levels. So active patients had a more diverse microbiome compared to inactive patients. The overweight, obese, inactive down here had the least diverse microbiome, and Faecalibacterium, a cancer protective genus, was more enriched among active cancer patients. So here, whenever you have inactivity, it was associated with reduced microbial diversity.
Can I ask a question about that?
Sure.
You said that athletes had decreased.
Yes. Yes. Now, athletes are a little bit different from the normal people, and that we can. But no, it's a very interesting question because it's not consistent. And I've done a lot of work on athletes and immune function, and realized many of the studies are super small. Athletes often have a very unusual diet. and they can be very stressed too. So I think like mostly they often have a very limited range of diet when they train, so maybe some of that is related to that. But perfect, yes, you got it already. All right, okay. So next question is like, what do these groups mean in terms of colorectal cancer survival? So Caroline looked at nearly 1,000 of our patients. And first of all, the less active patients, and these are like, this is a cut point that's at the higher end, but still important, metabolic equivalence of 18, had a consistently increased risk of death and recurrence. Likewise, overweight or obese patients had an increased risk of death and recurrence. And here you can see now these different groups. So this time it's the other way around. Highly active normal weight is the reference group. When normal weight patients are not active, we have a hazard ratio of nearly 3, threefold risk of colorectal cancer disease-free survival, so a risk of death or recurrence. And you can see even among the obese, compared to the highly active, the not highly active have a significantly different association, and with the worst survival outcomes for inactive and obese patients. There's, of course, the confidence intervals are getting somewhat larger, but nevertheless, it suggests that even when you're obese, activity can make a difference for colorectal cancer survival. So to close the loop, physical activity can counteract obesity-induced inflammation, and that maps with survival. So again, we're using our existing patients with our biomarkers. Physical activity was associated with inactive patients had higher CRP and IL-6 levels, although not statistically significant. But for obese patients, that is statistically significant with higher CRP levels, something that has been reported in the literature. CRP is just basically driven by body mass index. Now, looking at the combined associations, here's CRP and here's IL-6. Normal weight and active have the lowest levels of CRP, with 88% greater CRP levels when these patients are inactive. And among those who are overweight, obese, or inactive, you have the highest CRP levels, and when patients are obese or overweight but active, it's a significant reduction. So you can see that basically physical activity still had an impact on CRP levels, even among overweight or obese patients. So closing the loop again, the gut microbiome the question, is that associated with disease-free survival? This is work by Armin Bird and Madili Ilosumba. Armin is at Moffitt and Madili is a postdoc. This is a first study. We had only 166 patients here, but it goes in the consistent direction with greater alpha diversity being associated with improved disease-free survival. And then there was one specific genus that she looked at and a specific order that both seem to indicate signals. This needs to be replicated in larger patient populations, but at least it goes in the right direction. So going back to this pathway, I shared with you some of these different factors, and those were originally thought to play a role only in colorectal cancer risk. However, we have now also evidence that they matter in relation to colorectal cancer prognosis. Just to conclude, what have we learned about energy balance in colorectal cancer? Obesity is a major risk factor and prognostic factor for cancer. There are a multitude of biologic mechanisms linking energy balance as a black box to cancer and now to survival. If you want to, we can talk a little bit more later about these other mechanisms. And I also shared with you that adipose tissue is a key player systemically as well as paracrine as part of a tumor microenvironment, but that the type, distribution, and amount matter in relation to risk and the systemic inflammasome. This initial crosstalk may also give us opportunities for intervention, and that's what we're working on in partnership with Steve Hursting and colleagues in North Carolina. So, just to keep in mind, even though we're all busy, we should still try to help exercise and encourage our patients. Okay, let's talk about a different patient group, early onset colorectal cancer. So for many years, we observed nice decreases in colorectal cancer rates. And here, like age is 65 plus and age is 50 to 64. And then all of a sudden, this curve emerged here. And these were patients aged 20 through 49, not undergoing screening at that age, and their incidence rose. This increasing incidence has actually now more than doubled. You can see these are birth cohorts in the 1930s, those born in the 1960s. Those born in the 1990s have about a 2.5-fold increased risk of an early onset colorectal cancer. We don't know why. There are many hypotheses that have been brought up. The only thing we know, it's not genetics. It cannot be genetics because it's a change over a very short time frame. So there's a lot of research underway on diet, lifestyle factors, environmental exposures. Maybe it's the forever chemicals, maybe it's electronics. We don't know as of today. But we did a little bit of research on it. I want to share it with you today, and that was basically taking within our ColoCare study a multi-omics approach to look at what could be signals. First of all, Caroline and Sheetal Hardikar led work on early onset patients, showing that they were more often diagnosed at advanced stages, received more aggressive treatments, regardless of stage, which makes sense clinically, had lower rates of smoking, overweight, obesity, lower rates in our population of overweight, obesity. And then Andriana Holavati, who's faculty now at Vanderbilt, went out to look at the existing data we had on the transcriptome, metabolome, proteome, et cetera, and wanted to see, is there a unique molecular phenotype of sporadic colorectal cancers among young individuals? So here you see it's 233 patients. They were all microsatellite stable. So we tried to eliminate our genetically predisposed from an MSI perspective. And she found 312 genes differed in the tumor versus the normal mucosa tissues between these disease onset groups. And I'll share with you the multi-omics analysis results, which showed that an imbalanced redox status may be a distinct hallmark of early onset sporadic colorectal cancer. So what does this look like? Here's the transcriptome, where the alterations in NERV2-mediated oxidative stress response was standing out. For the metabolomics in panel B, glutathione metabolism emerged as a signal. For the proteome, we had no signals, but we also had very few patients in that group. And for the inflammasome, we saw decreases of systemic CXCL12. So Andriana said, well, this might all pull together in relation to the NERV2, master regulator of intracellular antioxidant responses. Now, this could be in relation to specific environmental exposures that we see this kind of oxidative stress response in our early onset patients. Of course, we also wanted to look at what happens to our ColoCare patients with early onset colorectal cancer. We did a survey of 202 patients to ask them about employment, insurance, financial outcomes, etc. And we tried to contrast what's happening in younger patients versus older ones. The younger patients were more likely to work after diagnosis, but they also had job lock. They did not want to move their jobs, neither did their spouse, because of worries about losing insurance. They also reported very much so higher levels of financial and psychological hardship. And I just want to share with you some of this data. So more than 35%, the purple group is the youngest, 18 through 49, took on credit card debt. Nearly 30% had to borrow money. More than 15% thought about filing for bankruptcy, and more than 25% each worried about money for household utilities or nutritious meals. So we have a major challenge that we have to help our younger colorectal cancer patients, and now I think also our younger breast cancer patients. So to summarize, early onset colorectal cancer is emerging as a major public health challenge. The etiologies are unknown to date. They must be environmental. Multi-omics suggests that oxidative stress, as vague as it is, really could be a part of this solution and pathway. And there are many challenges in cancer survivorship, and some of them I didn't even talk about, like reproductive toxicity. So we need to do more work on early onset colorectal cancer survivorship. All right, with that, I'm going to move quickly to something entirely different, which is exercise and the adjuvant therapy of cancer patients. I had done quite a bit of work in my past at the Fred Hutch, which was among healthy individuals. This is the National Center for Tumor Diseases in Heidelberg, where we did studies in cancer patients. Now, exercise generally shows benefits in the adjuvant therapy of stem cell transplant patients. And here's an example of a trial that Joachim Wiskemann did as part of his doctoral thesis. It was exercise prior to and during the transplant process. Here you see during the inpatient period. And these are the curves for cancer-related fatigue. The red curve is the control group, and the blue is fatigue among our patients who had regular strength training. And the greatest benefits were seen in the initially unfit. And here's the data on mortality after transplantation. It was not powered for that, but nevertheless, there is a suggestion of a signal that our stem cell transplant patients who were part of this exercise arm in this randomized trial did somewhat better. We did a number of studies also on breast cancer patients. I don't have time to talk about that right now, but for example, if you're interested in work of exercise during radiotherapy, it showed reduced fatigue and reduced pain, and biomarkers related to here seem to be IL-6, the IL-6 axis. Now, I'm going to tell you about a trial that's not published yet, and I know, sorry, for people on Zoom, the result slides I can unfortunately not have on Zoom because they're not published yet, but I will verbally talk about them. So this is a team at Huntsman Cancer Institute where we did a study called PEP study, Precision Exercise Prescription, and it's primary and secondary lung cancer patients undergoing surgery. I have a fantastic colleague here, Tom Varghese, who is a lung cancer surgeon who strongly believes in exercise being relevant for his patients when they come to have this highly invasive surgery. The exercise intervention studies that had been previously done in lung cancer were promising but limited. Many of them tested what we call like a one-size-fits-all model, so a specific prescription, which does not work in a clinical setting where many patients come in with a very different performance status, one versus the other. So you need to target and tailor. No cost effectiveness had been done. So translation into clinical practice had not yet been accomplished. Therefore, our aims were to test the clinical effectiveness of exercise, of a personalized exercise program in a surgical setting, and a program that's readily translatable into the clinic while generating the data needed for future reimbursement. We randomized 200 patients, and our primary endpoint was physical function, 6-minute walk distance at 2 months post-surgery. And here you can see the PEP intervention. It combined a remote as well as in-person implementation. We started in person during the post-surgery clinic stay. That was in consultation with a physical therapist. And why we call it a precision exercise prevention has to do with the daily assessment of the AMPAC score. It's a very quick assessment devised by physical therapy on how patients are doing from a functional level. And that precision component then resulted in adaptation of the program as needed. And this remote implementation then continued after discharge until 2 months. And our goal was here to really do weekly motivational interviewing. It was under psychologist's guidance, and it's an established way of working with patients to achieve behavioral change called MAPS. So this exercise prescription, again, was tailored with the MPAC based on functional limits and assessments were done at key time points. Here's the PEP study population. We had about 50% of them being low mobility in either arm, and randomization was stratified by this. I do want to say that thanks to an amazing surgeon, our recruitment rate was 87% of those that were eligible for the study. And as you may also see, we started this before COVID, and we ended afterwards. And we were able, thanks to the support by the Cancer Center director and by the physicians, to continue the study successfully during COVID. So here are some of the key results. We unfortunately did not quite meet our primary endpoint, which was the 6-minute walk distance, but the trend is absolutely in the right direction. Here's the exercise arm where we see an improvement. Here's the standard of care arm where we see a slight decrease. The p-value sneaks up against statistical significance, but not quite, because we had to do also, of course, an intent to treat analysis. We had very few dropouts. I'm sorry, I forgot to mention that. And here we see that, nevertheless, there are big differences. Women had a significantly improvement in 6-minute walk distance with the exercise program, more so than men, and older patients showed greater improvement. We did see statistical significance for our secondary endpoint, fatigue, at 2 months. So here you see in blue the exercise arm, where fatigue decreases over 2 months, whereas there's a major increase in our standard of care arm without the continued intervention. And again, women were benefiting more. So overall, to summarize, this program showed, as a clinic-aligned precision exercise intervention, significant improvements in fatigue and suggested improvements in physical function. We had very low dropouts despite COVID. We had only about 12% dropouts, and we had to, unfortunately, have our patients wear masks, of course. I mean, not unfortunately. Fortunately, they wore masks at the 6-minute walk distance during the periods of the pandemic, but that could have impacted our 6-minute walk test and distance that were only applied in some parts of the study and not in the other. So we're one step closer to clinical implementation. And so, again, another plug for doing more exercise. Okay, I'm going to spend no more than 5 minutes talking about Huntsman Cancer Institute. Here are our 5 buildings. As I said, we're 25 years old. Here's our recent retreat where we were celebrating our renewal, and I just want to give credit to an amazing, amazing team that is a pleasure to lead. Now, these blue dots indicate cancer centers across the United States, NCI-designated cancer centers. You see a lot of blue dots over on the East Coast. You see a few in California. And this is Huntsman Cancer Institute. There are no blue dots in those 5 states, or in these other 2, as we just talked about. But anyway, we made a decision to formalize our collaborations and interactions that we have with these 5 states, Montana, Idaho, Wyoming, Nevada, and Utah, to broaden what we call not our catchment area, but the area we serve. And the area we serve is now 10 million people, 43 American Indian tribes and nations, and 35,000 annual cancer cases. We have some affiliate hospitals here, but we also have now 10 senators and a number of other representatives that I actively try to convince of the value of research, and especially cancer research, on a continuous basis. And they're doing a good job listening. So one common theme across these 5 states is distance as a disparity. And it's hard to imagine here, but let me just read to you real stories. One of our patients lives in Price, Utah, about 2 hours from Salt Lake City. She's distraught because travel to HCI is not safe during the winter months crossing a mountain pass. She also suffers from diarrhea, and a long road trip without facilities is a near insurmountable barrier. Or last week, I saw a patient from Elko, Nevada, diagnosed with esophageal cancer. We scheduled his surgery, yet the remaining treatment plan is unknown. He owns a farm and does not have the time or capability to travel on a regular basis for further treatment. And I know these are situations that are similarly happening in the Central Valley and other locations where we need to reach out and care. By the way, when I showed this picture to Dr. Bertagnoli, our NIH director, she looked at it and she said, oh, this is where I grew up. So, Wyoming. All right. We, of course, don't have 1 billion locations, but we have a lot of partnerships across the Mountain West, and our COE office recently summarized these with little dots. We work a lot with federally qualified health centers, with American Indian partners, with schools, etc., and really try to make a difference. We also really make a concerted effort to listen, to learn, and to convene. We held multiple Mountain West Cancer Summits. Here's the most recent one from April, where we convened regional leaders, American Indian tribal leaders, and we jointly work to overcome distance as a disparity. This is one of our basic scientists who actually has taken a real interest in education about research to our CAB members and to others from the Mountain West. In terms of clinical trials, so now we have 5 states, right? The red circle here is 150 miles. Many patients travel more than 150 miles, 30% of our patients, and we're successful with enrolling them on clinical trials. 29 of our treatment accruals are from outside this, and the dark blue here indicates accruals of patients in these counties. So we use a number of strategies to facilitate accrual, including travel and housing subsidies, and more than 1/3 of our patients come from rural or frontier counties. We also have a trial portfolio that tries to test like short-term, short course radiation and other ways to address rural needs. We also try to reach out through Huntsman at Home. It's an oncology hospital at home model. It was published, and in the interest of time, I won't go through it in detail, but we have shown that this Huntsman at Home model results in reduction of rehospitalization, emergency department visits, and now most recently in rural counties, similar effects. So we are really trying to work on that. We're also hoping for an ARPA-H grant to do this work more thoughtfully. We use AI to think about how can we improve care over a distance. And here's an example of our investigators who wanted to predict symptom deterioration during cancer chemotherapy. So these patients leave our hospital, they're going back home, we're worried about them, and let's try to use AI to learn from the symptoms they report over the phone, actually, like with just pressing buttons, and see whether there is a future problem, and the model could predict future symptom scores for up to a week. So this is going to be one of our goals, to personalize symptom management in cancer patients over distance by using these kind of triage mechanisms. So, of course, none of this could have happened without many, many, many other people, and I am very appreciative of the groups at Huntsman Cancer Institute, at the National Center for Tumor Diseases in Heidelberg. That's my department there, and our ColoCare group, a lot of different grants, many of them still ongoing, a lot of wonderful collaborators in ColoCare. And I do want to put in a quick plug that we have a conference happening on transformation in cancer care delivery, where we have speakers from the NCI, from industry, from colleagues across the nation to think about how we can have new models in cancer care from screening to treatment, transform patient navigation, revolutionize prevention and cancer care delivery, and overall work towards making a difference together in innovation. So we also have poster awards for anyone who might want to submit one, and I'll send the announcement around. So with that, thank you very much for your attention. Looking forward to discussing.
I was wondering if you could tell us a little bit about the disparities in the colorectal study with socioeconomics and race.
So our ColoCare study currently has about 250 Black participants. And when you look at the locations, we have not, we have about 100 Hispanic participants. Clearly, these discrepancies happen. There has been a pilot initial analysis, and survival looks like, even though we are like major cancer centers, we still have reduced survival among our colorectal cancer patients that are coming in. We do not see differences for rural frontier versus urban, and maybe in parts because we didn't do such a concerted effort to try to help them out. So yes, still discrepancies. Okay, maybe it's for.
As you look at gender effect, we looked at C-reactive protein as an overall predictor of all cancers. It only played a role significantly in lower income women.
Yeah, well, this is all adjusted for sex, and we also did multivariate, so it's multivariate adjusted, and we also did stratified analyses. We did not see a difference between men and women in this particular study, at least not statistically significant or significantly worth of reporting for colorectal cancer.
Second, having all this great data on inflammatory effects, have you looked at cardiovascular disease? Because we're seeing in our endometrial cancer patients more dying of cardiovascular complications after 5 years than dying of cancer.
I think that's an excellent question. Yes, like Jane Figueredo from Cedars has started to do that analysis. We're trying to get a grant to not do all the work for free with the UO1 funding, I should say, provides only funding for the basic infrastructure. It's only for collecting the patients and following up the patients. It's not supporting any science, so we need additional R01 funding or other, free labor if we're lucky to do the research. But we've been able to do quite a bit. We're trying to get funding for a larger grant that looks especially at outcomes for early onset versus later onset, and the factors that you mentioned. Like there's a multi-dimensional model for toxicity, which includes physical toxicity, mental toxicity, socioeconomic toxicity, and cardiometabolic outcomes are part of the physical toxicities.
Have you planned to incorporate germline genetics in any of this, specifically trying to look at, for example, sort of the microbiome stratified by BMI, let's say stratified based on something like the BMI polygenic risk score, to try to disentangle those things a little bit, and reverse causality, I think, is still a concern there, even when you have like pre-surgical samples.
So the question for those online was like, to what extent we plan to integrate BMI, not BMI, genetic factors on stratification. I do want to say that our goal is first to use what we can actually measure, right? Because like measuring an actual biomarker or measuring an actual body mass index or a fat distribution, in my view, is a little bit more valuable than inferring it. But we do have, genetics being done on a subset of our patients. They're part of the GECKO consortium, so we are contributing the data to all the large analyses that are happening there. But we should probably ask them to get the data back to do some analyses just for the fun of it too. So thank you, good suggestion. Yeah.
So it seems like since you have such holistic research going on, that I could probably find 20 things I'm supposed to do in order to reduce my cancer risk by 5%. If I do all 20 of those things, I'm never going to have cancer. But at the same time, because of the interrelations and diminishing returns and all those kinds of things, that's really what I want to know as a layperson. How do I manage what I can do in order to reduce my risk?
Yes, and I think it's a critically important question, and then cancer is not equal to cancer, right? So for colorectal cancer, we know that there are a lot of factors that can be playing a role in prevention, especially like physical activity and obesity are very strongly associated with an increased risk of colon cancer. For other types of cancer, for prostate cancer, for example, there is less strong of an association with environmental factors and a stronger association with hormones. So cancer is not equal to cancer. I can tell you two things, like from a cancer prevention perspective. First of all, we know smoking is the number one risk factor. It's relating related to 16 types of cancer. Most people don't know that. They just think it's only lung cancer. Even colon cancer is elevated with smoking. Obesity and lack of physical activity is the next large block of cancers. 13 types of cancers are elevated because of obesity being growing and physical activity not being at the levels that it should be. So these are the two biggest blocks. Most of what I talked about is about survivorship. When cancer patients ask us, what can I do to improve my chances of having a happy, healthy life and avoiding recurrence? And so we've done work on diet and on, here, like I presented, energy balance. So it's a slightly different angle here, but very important question. Thank you.
Great talk. This epidemic of young people with colon cancer is a real problem, as you indicate. And it looks like a diet effect. If you look at other cancer types, how people in other parts of the world have a higher risk of stomach cancer. They move to the United States. The stomach cancer risk goes down with moving to the United States with changing diet. It looks like a change in diet to me. And I wonder, people who really think deeply about diet, are there specific hypotheses, given like what's come into our food chain over the past 20-ish years or so, that is it high fructose corn syrup, or is it, are there specific candidates that can explain this?
I wish I could say that. My background is actually in nutrition. And so whenever we have a hypothesis, there's something that shows, it may not be true. And it's, Germany has not the same level of processed foods, shall we say, or not the same changes in processed foods or high fructose corn syrup in particular, and we still have this rise. We definitely see it more in the Western countries, but now we also have breast cancer coming up, and breast cancer is typically more hormonally associated. So I think, there's a lot of work we need to do. There may be a challenge with some forever chemicals and some other plastics and other substances, but then again, Germany doesn't have as many plastics. So why do we? But we need to figure out. Again, we see this oxidative stress in the middle there. So maybe it's worthwhile to try to navigate from the other end. What causes maybe this oxidative stress? And we're having so many great research studies underway right now and analyses that within the next 5 years we should have some pretty good answers. But they're like prospective cohorts, and they still are measuring. Yeah.
Great talk. So we are doing a pretty good job, I think, of screening, and especially in those older age groups. If we change the screening, because there's where we pick up polyps, which also impacts the cancer incidence. If we screen a lot better, do you think that we would also reduce the risk? I'm sure that there's other risk factors like diet and other things that we may not know about, microplastics and things that people are exposed to. But if we shift the screening, that could have some effect in terms of reducing the incidence of mortality.
Yes, great thought. And the screening guidelines have been lowered now to start at age 45. It's still always a question of cost effectiveness. It's still a rare disease among these young patients, and there are risks associated with colonoscopy. So maybe they should do more regular FIT tests and other tests as instead of a colonoscopy at that level. I just want to say, it's like, and I had discussion this morning, it's like not the people you expect to get an early onset cancer is a perfectly healthy, exercising, slim person coming in with a colorectal cancer at age 35 with no family disposition.
Cancer is increasing in younger people. We've got a lot of work to do.
Yes.
So, just want to tell everybody there's a reception on the patio, and please come and enjoy.
Yes.